Hello students, I am Dr. K. Sarojini Chakravarti, working as Assistant Professor in the Department of Botany, Government Degree College, Khairtabad, Hyderabad. In the last class, we have seen what is fungus and what is the mode of nutrition in fungus. So, the mode of nutrition in fungus is three ways. One is saprophytic, parasitic, and symbiotic. Why fungus has these kind of mode of nutrition is because they are micro or macro organisms and they are heterotrophs. Heterotrophs means they cannot prepare their own food and they depend on other organisms for their food. So in this way, to derive nutrition from other organisms, so fungus it adopts three ways of modes of nutrition. So among the three ways, we are going to deal with parasitic fungi. Okay, in parasitic fungi means it depends on other organisms and derives its nutrition. So depending on the type of host, whether it is a living host or a dead host, again it is of two types. One is biotrophic, where it requires a living host. Another one is necrotrophic, where it kills the host cells, and again from the death of the cells it derives from nutrition. So first fungal disease, plant disease, what we studied is white rust of crucifers caused by albugo. So last class we have seen vegetative structure of mycelium of albugo, and what is the mode of nutrition symptoms, white rust of crucifers, and mode of what is the reproduction, mode of reproduction, that is asexual and sexual. So we'll continue the class with asexual reproduction in albugo. Asexual reproduction in albugo, it takes place in three steps. The first one is formation of conidiophore or sporangiophore. For that is first one. After that, formation of conidia or sporangia or zoosporangia. That is the second step. After that, germination of sporangia. So the germination of sporangia, it occurs in two ways. So depending on the temperature, it may be a direct germination or it may be an indirect germination. Now we'll see each step in detail. So in this picture, this is complete asexual reproduction in albugo. Okay. So what is the first step? The first step is formation of conidiophore or sporangiophore. So for this, what it has to do is albugo, the fungus, it has to enter into the host tissue. Okay. So after entering into the host tissue, it develops, and what type of mycelium it is? It is intercellular mycelium. Okay. So the intercellular mycelium, it develops beneath the, so this is the host leaf. Okay, so through any stroma or stomata, the pathogen, a fungus, it enters into the host tissue and it starts, developing. Okay, so the type of mycelium growth is intercellular mycelium. So in between the host cells. Okay, so it develops a mat-like structure below the lower epidermis of the leaf. Okay, so after that, from this mycelium, the tips of the mycelium, from the tips of the hyphae, you can see development of short, erect shape, thick walled, club shaped structure known as conidiophores or sporangiophores. Okay, so this is the host tissue, and the type of development is intercellular growth and below the lower epidermis, and this is cenocytic mycelium, and it develops below the lower epidermis and it's form a mat-like structure, and from the hyphal tips you can see development of short, erect, club-shaped sporangiophores or conidiophores. So they are nothing but the stalks. The stalk is known as from where the sporangia or conidia develops, that is known as either sporangiophore or conidiophore. Okay, the shape is club shaped. Okay, so after that, what happens? The apical end of this conidiophore. So this is the apical end of the conidiophore. It is multinucleate. Why it is multinucleate? Because the mycelium is multinucleate, cenocytic. So that's why The sporangiophore or the conidiophore is also multinucleate, and it is densely cytoplasmic. So you can see dense cytoplasm at the apical end of the conidiophore. So with this, the first step has come, means entry of pathogen albugo into the host cell or host tissue has occurred, and development of mycelium has occurred below the lower epidermis, and it has resulted in the formation of short, erect, club-shaped sporangiophores or conidiophores. Right. Next, it enters into the next step, that is formation of conidia or sporangia or zoosporangia. Okay, why these three? Okay, so there is a variation. Okay, so from the tips of this conidiophores or sporangiophores, if conidia develops, it is known as conidiophore. If sporangio develops, it is known as sporangiophores. From this tip of the sporangiophores or conidiophores, you can see long basipetal chains of conidia or sporangia development takes place. Basipetal chain. So what is basipetal manner? Basipetal manner means the mature one is at the below, and the mature one is at the below, and the young one is at the below, and the mature one is at the top. So that's why you can see the chain is, see, smaller ones are near the hyphallens or the of the conidiophores or sporangiophores, and the mature ones at the top of the chain. That's why it is known as basipetal formation. So how it forms? So during the formation of conidia or sporangium, a deep constriction. So you can see a deep constriction. Okay, a deep constriction appears below the swollen end of the conidiophore, which generally increases in size. Why? Because there is an inflow of cytoplasm from the tip of the conidiophore, or from the stalk of the conidiophore, sporangiophore, to the tip of the conidiophore. So it bulges, it increases in size. So after that, ultimately, a constriction develops, forming a transverse wall. Okay, so with this you can see formation of a spherical multinucleate conidium or sporangium. Okay, so this process is repeated several times in such a way new conidium or sporangium is formed on the conidiophore or sporangiophore in a basipetal manner. Means, as I said, youngest one is at the base and the mature one is at the top. Okay, so that's why it is basipetal. And in between each conidium or each sporangium, you can see an intercalary disc. Okay, see, it is also known as disjunctor. Okay, why it is known as disjunctor? Because this disjunctor, it forms a common wall between the upper conidium or upper sporangium and the lower sporangium. Okay, so it forms a common wall. So when all these are intact, the chain of conidia are intact. But because of this presence of this disjunctor or intercalary disc, when what happens? So when this chain of conidia, it exerts pressure on the epidermis of the host cell. Okay, when it exerts pressure on the host cell, so exert pressure means, so when you're pushing something, what happens? The elasticity of the particular when you drag an rubber band or elastic rubber band from two ends, so what happens? It creates one kind of tension, and at the center it breaks into two. The same way, so this conidia, the chain of conidia or sporangia, it creates some kind of pressure on the upper epidermis. So when it creates a pressure on the epidermis, so it, with this, there is a tension in the epidermal layer, and it splits open or breaks open, and all the conidia or sporangia will be exposed out. Okay, so when they exposed out, the color of the conidia is almost shiny white color. Okay, so when they are exposed out on the epidermis, lower epidermis of the leaf, it appears as white powdery mass. That's why the disease is known as white rust. That's why the disease is known as white rust. So each conidium is round. Okay, it is smooth, multinucleate and spherical in structure. Okay, so what happens when they are liberated out? So this disjunctor or this intercalary disc, it gets dissolved between the two conidia, and when they are liberated out, so all these conidia will be dispersed in, and they are dispersed out. So individual conidia will be liberated out, not in a chain. That's why intercalary disc is important. So this intercalary disc, it allows the conidium to be get separated from each other when they are exposed out from the host epidermal layer. Okay, now from this it enters into the third step, that is germination of conidia. So as I said, the germination of conidia, it depends on the temperature. Okay, so depending on the temperature, it occurs in two ways. When there is a high temperature, means when the weather is dry, the whatever sporangium is there, it acts as a conidium. Okay, so conidium is non-motile in nature. It acts as a conidium, and it is non-motile in nature. So this, with the help of wind, okay, it is liberated, and when it falls on a suitable substrate, that is any host tissue, again it starts its germination. Okay, so it starts its germination by forming a germ tube. So this is a germ tube, so this is direct germination by conidium. So this is the stoma of the host tissue or host cell, and this conidium, when it falls on a suitable substrate, so from this conidium you can see development of a germ tube which enters into stoma, and again its life cycle, asexual reproduction starts, by developing again a mat-like dense mycelium below the epidermis. Okay, if the weather is not dry, if it has low temperatures, and the weather is humid in nature. Okay, so when the weather is humid in nature, when temperature is humid and it is not dry and low temperatures are present, then this conidium, it acts as sporangium. It acts as sporangium. So now this multinucleate sporangium, okay, so this multinucleate sporangium, it divides into a number of uninucleate segments. Okay, each segment. Okay, so what is segmentation? Means you can see a nuclei around the nucleus. You can see it is surrounded by protoplasm. A nucleus surrounded by protoplasm. Nucleus surrounded by protoplasm. So this is known as segmentation. Okay, so each segmentation, nucleus along with the protoplasm, it metamorphoses or it changes it into reniform biflagellate zoospore. Okay, so reniform biflagellate zoospore. Okay, so this reniform biflagellate zoospore, as it has flagella and biflagellate, and it has two types of flagella. One is whiplash, another one is tinsel. So as it has flagella, it can swim. It will swim in the water. Okay, water droplets. Water is not water. Water droplets. Okay, so water droplets is it swim for a while and it take rest. Okay, so after taking rest, it withdraws its flagella. It will be encysted. Assist means a thick wall is formed around itself. So this is known as encysted zoospore. This form is known as encysted zoospore. Now this encysted zoospore, now it forms a germ tube. So this is a germ tube. So with this help of this germ tube, again it enters into the host tissue through stomata, and it penetrates into the host epidermis and develops into again intercellular cenocytic mycelium. So this is asexual reproduction in albugo. So asexual reproduction in albugo takes place in 3 steps. One is formation of conidiophore or sporangiophore. The second one is formation of sporangia or conidia, and last is germination of conidia or sporangia. So depending on the temperature, it may be direct way, that is through conidium when temperatures are high, or it may be indirect way, that is through zoospore formation. So this is asexual reproduction. In the next class, we'll continue with sexual reproduction in albugo.